Bhujbal Knowledge City, the part of Amity League of Colleges, driven by a single-minded focus on imparting quality education. Amity BKC Institute of Pharmacy established in 2006 with a mission to develop students socio-technically. The institute has maintained the quality right from the establishment, as it reflects from the accreditation and various awards which college has achieved. Amity IOP is the first NBA accredited pharmacy institute in Nasik region. Amity BKC Institute of Pharmacy has been approved by AICTE PCI and is affiliated to Savitribai Phule, Pune University. Amity BKC IOP offers full-time courses in Bachelor of Pharmacy, Masters of Pharmacy and PhD. Institute stands for its excellent, well-lighted and ventilated ambience with range of facilities like classroom which are equipped with an overhead projector, speakers, smart board, etc. Well-equipped and spacious laboratories which provide experimental learning experience. State-of-art pilot plant simulating the pharmaceutical industry with range of equipments for formulation, development and evaluation. Institute has separate instrument room having range of sophisticated instruments as per the need of industry and research work like HPLC, UV, FTIR, etc. Heart of Institute, the library, one of its own kind with open access, great ambience and various national and international book collections to give great learning experience. Institute is also a well with gymnasium which is equipped with a number of professional electronic and manual universal fitness machines along with indoor sport facilities and music room. Campus also has an amphitheater for social gathering and expression of students. The institute believes that the quality of faculty member and their commitment to excellence is the core competence towards its development. With this thought, institute have more than 50% doctorates, 200 plus publications, 5 patents, 1 copyright and 16 book publications. Our students are encouraged to take part in national and international competitions and events as part of their curriculum. To add feather on hat, we have Ms. Archana Kapse, Air 3 in GPAT 2019, Mr. Aditya Darade, Air 1 in GPAT 2014 and Mr. Parag Mehta, Air 15 in GPAT 2010, who have qualified national level exam GPAT with dignified ranks. Amity IOP has signed 8 Memorandum of Understanding, that is MOU, amongst them are Japanese Industry, Asahi Kasei and Malaysian University Technology Mara. Amity Institute of Pharmacy provide a platform for expressing students and showcase their hidden talent through Antaranga and Amity Utsav annual social gathering. Success comes down to you with appropriate attitude, effort and focus, which is certified by various achievements which institute has received. Best Magazine Award by SPPU, Champions Trophy at National Pharmacy Week, Award for the Best Research Paper and Presentation, Best College Award by Student Development Board SPPU for Student Welfare Activities. Institute has also been honored with highest rank Platinum by AICTE C2 survey for its outstanding linkage with industries for consequently 2 years, which surely strengthen the institute and students to survive in this brave new world of technology. Bhujbal Knowledge City Institute of Pharmacy, the place where your search ends.
Hello, Umesh sir. I am audible.
Yes, sir, you are audible.
Okay. A very good morning to one and all. Myself, Pratap Pawar, welcome you all for a national level one-day seminar on herbal drug industry, mind to product, for pharmacy aspirants, organized by IQAC Cell and Department of Pharmacognosy, Mumbai Education Trust, Institute of Pharmacy, Bhujbal Knowledge City, Adgaon, Nasik. It gives us immense pleasure to welcome the today's first speaker, Mrs. Anjali Kataria Madam, and respected Dr. Sanjay Shirsagar Sir, Principal of Meds Institute of Pharmacy, Bhujbal Knowledge City. The Meds Institute of Pharmacy is making a 30 years of excellence with the generous support and under visionary leadership of Honorable Sri Chagan Bhujbal Sir, Sri Samir Bhujbal Sir, Sri Pankaj Bhujbal Sir, trustees of Mumbai Educational Trust, and respected Dr. Shefali Bhujbal Madam, the Chief Administrator of Bhujbal Knowledge City. The institute has started its journey towards academic by excellence in 2006 and it's approved by AICTE, PCI and affiliated to Savitribai Phule Pune University. MAT IOP is a NBA and NAC accredited and is honored with highest platinum rank in CII survey for industry institute interaction by AICTE. The institute stands in the band of 76 to 100 pharmacy institutes across the India in NIRF survey in 2020. The Meds Institute of Pharmacy have various courses like B Pharmacy, PharmD, M Pharmacy, in pharmaceutics, in pharmaceutical chemistry, quality assurance technique, and in pharmacology. It is also SPPU recognized a PhD research center. With the beginning of MAT is redefining the system of education to meet current need of market even in pandemic situation of COVID also. Now, the today's seminars is focusing on the different pharma aspirants to give the idea about herbal industry, startup, how to start the herbal industry, the setting of different herbal formulation on industrial scale, and their regulation aspects of herbal industry, and development and manufacturing of herbal products as well as Ayurvedic product. So with this brief today's introduction, now I request the today's convener Dr. Sunita Surse Madam, HOD of Department of Pharmacognosy, to give the brief introduction about today's event. Surse Madam, over to you.
Thank you, Pratap Pawar sir. On the occasion of this national level one-day seminar on herbal drug industry mind to product, I, Dr. Sunita Surse, welcome all the respected dignitaries which are present on this virtual platform. Our eminent speakers, Mrs. Anjali Kataria Madam, who is co-founder of Rebe Pharma Sinnar, Dr. Shishir Pandey sir, director from the Ayurvedic Seva Sangha Nashik, and Dr. Sanjay Tamuli sir, CEO of Abhinav Healthcare Mumbai. I also welcome Honorable Dr. Shefali Bhujbal Madam, Chief Administrator of Bhujbal Knowledge City, respected Principal Dr. Sanjay Shirsagar sir, my colleagues, and all the participants who are attending this virtual seminar. Dear all, due to this COVID-19 pandemic, we have all gone through a difficult situation at all the levels. But at the same time, it has opened many new business and research opportunities for us. Entry of many newcomers to the industry as well as attempts to carve their niches to control market share can be witnessed in the coming years. With the current global trend towards these healthy products, people are turning more and more towards herbal and organic commodities. The global herbal medicinal product market, it is expected to witness robust growth in coming future. This growth is expected to be primarily driven by shifting trend from conventional medicines to traditional medicines, favorable government regulations, and growing production capacity of manufacturers across the globe. Considering these business opportunities and for facing new challenges, we, Pharmacognosy Department of MIT's Institute of Pharmacy and IPSC Cell has organized this virtual seminar where all manufacturing, regulatory, and startup aspects will be discussed and guided by our eminent speakers. Our first session speaker, Mrs. Anjali Kataria Madam from Rebe Pharma Senior, will guide us on herbal industry startup and share her own experiences with you. Dr. Shishir Pandey sir, who is director of Ayurved Sanshothan Vibhag of very old and well-known Ayurved Seva Sangha Nashik, he will highlight on development of Ayurvedic drugs, its manufacturing and analysis. The regulatory formalities and drug development process will be taken care by our third speaker, Dr. Sanjay Tamoli sir from Abhinav Healthcare, Mumbai. This seminar, it will be really helpful for those who are in research activities, especially herbal research activities, aspiring for herbal industry startup or planning for commercializing their herbal products on the large scale. I appeal all the participants to attend today's seminar, ask your queries to experts, and start your journey of herbal formulations which are still in your mind or maybe in laboratory on small scale towards the commercial products. Thank you, respected Principal Sir, for showing faith and giving this opportunity to me as a convener and our department to arrange such a novel seminar. Thank you for your idea. I request now Mr. Pratap Pawar sir to continue with the seminar proceedings. Thank you, sir.
Thank you, madam, for briefing about the today's speakers and the seminar. Now I request to our respected principal, Sir Dr. Sanjay Shirsagar sir, to present the keynote address.
Thank you, Pawar sir. At the outset, let me welcome all the delegates, participants for this one-day national workshop related to the herbal industry startup, that is Mind to Business. I welcome all the eminent speaker for today's workshop who have spared their valuable time for guiding the new generation and aspirant with respect to the herbal startup. Our today's first speaker, Dr. Mrs. Kataria Madam, who have set an example with her organization related to the herbal industry. She will start with the today's session. I also congratulate Dr. Surse Madam, convener of today's program, for organizing successfully or making proper arrangement with respect to the program, as well as coordinator Mr. Pratap Pawar for organizing this seminar and coordinating the activities related to this particular workshop. Moving to the today's topic, which our department have selected, and as we know, our nature have presented ample of opportunity for entrepreneurship by providing herbs. that is the department, pharmacognosy department, or our herbal, our nature, rather than department, I can say that our nature have given us the ample of opportunities in terms of herbs, in terms of certain Ayurvedic concept, which can be utilized in the best possible way to start our own business. And with today's workshop, we could able to understand how we can explore these opportunities, because many of us have some idea in our mind with respect to the herbs, because many of the home remedies are available, and many of us have an idea, but we don't know how to convert that idea into a successful business, or to how to start our business with respect to our idea. And as I have said, the herbal industry, or sorry, our herbs have given us a ample of opportunity to start the business. And we have today we have three eminent speakers, Mrs. Kataria, then Dr. Pandey, as well as Dr. Tamburi, who have set an example with their work in the herbal industry, and they will definitely share their experience. They will guide us with respect to how we can start our business with respect to the with respect to the herbal product. So it will be the great opportunity, which will be provided by the Department of Pharmacognosy and the IQCC, and I hope it will give the new directions to your ideas. So once again, congratulate Department of Pharmacognosy and welcome all delegates to the Met Institute of Pharmacy. Thank you very much.
Thank you very much, sir, for your precious word, for being motivational support to conduct such type of programs and event for the well-being of students and also faculty. Now here I again request to Dr. Sanjay Shirsagar, sir, please inaugurate the today's one-day national level conference of herbal drug industry, mind to product on digital platform. So please, I request to you.
Yeah. Thank you. So I declare that I have inaugurated
10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Thank you, respected principal, sir. And now I hand over the session. Now I hand over the session to Dr. Moreshwar Patil, sir, to start the first session of the today's seminar. Moreshwar Patil, sir, over to you.
Yes. Thank you very much, Pratap Bhavan. Good morning, one and all. Myself, Dr. M.P. Patil, welcomes you all for joining this one-day national level seminar on herbal drug industry, mind to product, organized by IQAC Cell and Department of Pharmacognosy, Meds Institute of Pharmacy, Bhujbal Knowledge City, Nasik. We are very fortunate to have with us Mrs. Anjali Kataria, madam, who is a co-founder of Reve Pharma, Sinnar, and I got an opportunity to introduce her. Madam is born and brought up in Nashik. She has completed her graduation in 1999, Master of Business Administration in the Specialization Systems from Pune University in 2001, and post-graduation in Quality Assurance Techniques in 2014. Madam started her career as a partner and QC/QA manager at Cytec Laboratories Private Limited, Musalgaon, Nashik, and worked for 8 years. Reve means a dream, and Reve Pharma is a dream comes true. So it was a dream of two professionals, Mr. Milind and Mrs. Anjali Kataria, who came together after a rich experience to establish a company which is manufacturing a standardized Ayurvedic products. Today, Reve Pharma is spreading its wings in the different segments like herbal cosmetics, herbal oils, herbal creams, dietary supplements under the leadership of Mrs. Anjali Kataria. They are manufacturing more than 900 formulations. The prominent, or we can say the key formulations development includes sugar regulating formulas and curcumin related formulas. Revia Pharma also manufacturing herbal cosmetics for renowned brands like McAfee and Mamaearth. They are one of the Ayurvedic manufacturing facility having their own instrumentation laboratory as well as in-house extraction facility. So with this brief introduction, now I request Anjali Madam to please enlighten the audience with her words of wisdom on herbal industry startup. Madam, please.
Thank you, sure, sir. Am I audible?
Yes, you are.
Okay. Thank you. First of all, I am so happy to see the interest of pharmacy college in herbal industry, herbal products, because 20 years back it was a different scenario. So I really congratulate you all that you have a great management, Honorable Shifali Madam. And so, respect with all your permissions, I would like to share my presentation first. Just excuse me if I am not very easy with this online thing. Okay, so is my screen visible?
Yes, ma'am.
Okay, here today is inspiring all these pharmacists, all these budding entrepreneurs for to be entrepreneurs in herbal industry. And even if I am able to inspire one of the crowd, I will be finding myself very fortunate. Yeah, if I am supposed to inspire you all, I have to speak about my journey every now and then. So yeah, it was a great introduction. Herbal, like Sunita Madam told in her introduction. So why people are going for herbal? So since this is a online platform, we need to be one way. So when allopathy introduced, allopathy was introduced maybe in early 1800s, that is 1820s, that is the 19th century. Means it is 200 years old. The science of pharmaceutical medicines is 200 years old. But how old is Ayurveda? Now, any people will have different answers, but everybody is right because no one knows the exact number of years. But it is about 5,000 years old ancient wisdom, because uh, all our rishis, all our old India has the richest, wisdom of this medicine and the most ancient. So that's why it is the oldest one, and that's why everybody in the world is going towards herbal. Now I would like to share our story here. Like Mureshwar sir said, sir has introduced, me and Milin, my husband, we started our career in 2002 with a small facility to produce cosmetic products. And since it was our own small endeavor, there was always a limitation of resources. We were aspiring for growth, and as every pharmacist wants to make medicines, we built a factory which was good for making pharmaceutical lotions and creams, ointments. We had also collaborated with a US company, and that day our American partner was going to come to visit the factory. Kaise banaya hai, let's see. And after going about the whole factory, he suddenly said, Milin, Anjali, why don't we go for Ayurvedic license? And since he had a great experience in the global business, uh, he knew that there was this opportunity of uh, doing good business in Ayurveda, which herbal. Herbal is a Western version of Ayurvedic medicine. It's just a name. So this was a great opportunity, and we decided to take a plunge in this new area for us, in spite of being pharmacists. Then let me tell you how much India contributes to the world herbal market. If you can see the number on my screen, 83 billion US dollars is the 2019 figure of business in herbal medicine, herbal market. And Indian contribution is only 0.5%. Why? Globally, why not Indian companies, Indian manufacturers, why they cannot go global? I would have liked to ask this question in the audience, but since what happens, we all make good medicines, we have a great knowledge, we have so much to share with the world. Since it is Indian, still we cannot go there and be a part of this whole big competition. But what kind of documentation is asked for? They ask for cultivation time, means what time when was this cultivated, what season. Then they ask harvesting time, means we need to document what time this was harvested, how old was the shrub or herb or tree or whatever. So any part of the plant, the activity. Did you know that chanting some mantras and this lunar eclipse and all these things, harvesting during these times definitely affect the activity of the herbs. And by the way, in our factory, constantly, continuously, we are playing this Mahamrityunjay mantra, and with only with the intention to put in positive vibes and all that positive energy in our medicines. So coming back to documentation, another documentation is required is how much actives is present in these herbs, means what kind of chemical is present, is it really present. And after that comes stability, means right now while harvesting you had this much of curcumin, but how much time is it stable, and can you able to prove that? So all these things are asked in documentation where we lack. Then comes an important point of standardization. बहुत बहुत सुना है ये ना word. This word is over. Both of us, me and Milin, we don't like to sit quiet. So in 2008, we started this new enterprise called Rave Pharma. It is not Reve, it is Rave, which is a French word which means dream. And we dreamt of this ancient wisdom being converted to modern science, modern medicine, and sharing it with the world. That's what is our mission about. And in our journey, few years down the line, when we realized that we could not control the quality of ready-made herbal extracts that we were getting. Some viewer, some attendee had asked this question of how to control raw material, some Amit Roy or something. So this is your answer, Amit. what happens, we cannot control the quality of incoming herbal extracts. So what we did, we decided to go backward. We did backward integration and put up our own in-house extraction plant. And then we could control that incoming material quality. But then what happened, the checking of this purity of our products, of our raw materials, started becoming a challenge. Then we invested uh, in these high-end instruments of HPTLC and atomic absorption, which helped us do the fingerprinting and also the testing of heavy metals. Because of these heavy metal testing, we can easily sell in the uh, bigger countries, bigger markets. So here is when we learn to standardize our standardize and document our products. So now you can say that we found the niche in the herbal industry where we can overcome this global competition. Now, speaking about the global competition, how much big it is? Currently, as I told you, 83 billion US dollars, that is 6 lakh crores in Indian rupees, is the 2019 figure, and it is expected to grow to 550 billion dollars. कितने zeros होते वो भी अपने को ढूंढना पड़ेगा. It is like such a big number. Means look at the scope. We'll look into this global scenario a little more. I would like to tell you that our herbal medicines, that is the Indian Ayurvedic medicine, goes as dietary supplement in the US. And earlier, means about maybe 4 or 5 years ago, India was only about psyllium husk, psyllium seeds, and senna bark, senna leaves. India used to export only these two products. I mean, it was known for majorly these two product exports. In 2018, we were attending this supply side west in the states. This is the biggest herbal dietary supplement exhibition, expo, or trade show you can call, that happens in the US every year. And we happened to visit that trade show. And as soon as we entered the huge space, what we saw was some same banners everywhere called KSM 66. KSM 66 was seen everywhere. And when we took a closer look at it, we found that KSM 66 was written as world's best ashwagandha. And it was such a proud moment. It was such a proud feeling to see that an Indian herbal company is sponsoring such a big show as a sponsorer. Also, it gave us a great feeling about such a big opportunity lies behind this. Means just they were just like our dream, like they have modernized this Indian legacy of 5,000 years of ashwagandha's knowledge, and they put it up as world's best ashwagandha. With this, I would just like to share a video with you all. Is my screen visible, sir? What did we see in this video? Sir, am I audible?
Yes, madam.
Okay, you could see the video. It was clear.
Yes, yes, yes. We have seen. We have seen.
Okay, okay. So can I ask these questions right now? I want to just see what people have seen in this video. If I could get some responses from the audience.
Yes, yes. Audience, please answer the
Okay.
question. You can answer the question
Anything.
in the chat box.
I'm not able to see the chat box, so you'll have to tell me what answers have come. Okay, so let's not waste time because I don't know how much time it will come. Uh, First was the harvesting time. When we started talking about documentation, harvesting time was one of the important things. If you people have observed correctly, the hunt is now rich in its active constituent. If you have observed correctly, the farmer lady who was sitting with the ashwagandhas, twigs, and the plant parts, she was wearing mask and gloves, all those things. So this is good agricultural and cultivation practices that was followed. Means this is also documented. Then they have proven the purity of ashwagandha by checking the vidaferin content. After that, they told how useful it is for the body and mind. And in order to prove these effects, they have done the clinical studies to uh, register the effects and to prove it and to make papers on all those things. So, clinical studies was another part of it. After doing all this, they are selling ashwagandha as good as an API, means active pharmaceutical ingredient in the US market. Today, it is a 700 crore product, single herb ashwagandha. The shelf that you saw there in the video, I've seen it myself and it was really proud to see that. So, this is the scope from only one single herb point of view. There is so many branches or so, like when I talk about scope, there are so many things that you can do. Means if at all anybody of you wants to become a herbal entrepreneur, there are so many options to choose from. Let me name a very few of them. Like one is organic farming. In organic farming, what we can do world over, especially in Europe and UK, organic organically grown ingredients or herbs are in such a huge demand that you can sell regular in you can sell an organic item three times its price against the regular non-organic material. So this is the wide scope of business in only organic farming. World over, even if I talk about this ashwagandha people, they have 1500 acres of ashwagandha growing organically. So we can imagine how big is the industry. Then there is vertical farming. What is vertical farming? I think it is heard by everyone, and most of them know it now. But I would just like to elaborate it a little bit. Vertical farming is nothing but you multiply the space by increasing the uh, mm, number of number of plants planted in one per square meter area. So I've seen this curcumin, that is the haldi, being produced in vertical farming, and the quality of turmeric was also great. The quantity produced was 200 tons in just a quarter of acre in area. Quarter area. Quarter acre area. Can you imagine that? 200 tons of turmeric in this much in one single season. And this turmeric was found to have as much as 7.5% of curcumin. This is just one example of one herb of vertical farming. So many herbs can be done. Then again, this single herb means this is this is taken from the example of ashwagandha, that is KSM 66. That is a big example. We don't we don't know how much is there in which ingredient. We can start with any herb, any common herb. Take amla, ginger, neem, anything which is available abundantly in and around the place. Actually, when we study pharmacognosy, all these names, those botanical names and all those things don't really interest you. Glycer as a glabra, as a directa indica, and embolica, ribes, embolica officinalis. I mean, all these things seem very boring during that time. But now you can see the other side of it. Vedania somnifera. This name was so very boring, but what they have done to it. KSM 66 ashwagandha, 700 crores. So you have seen the other side of it now. You this scope cannot be ignored. There is so much to be explored in Ayurveda. For example, amla. Amla contains around hundreds of 100 active components. But what we talk about? Only vitamin C. Vitamin C in amla does this, does that, does this. But there is a lot of activity to be still explored. Then again, another example I would like to share. Before that, there are so many antioxidant herbs. Some antioxidant herbs work as neuroprotective. Some antioxidative herbs work as rejuvenating, anti-aging. So there is so much to be explored. Another example is of turmeric. Curcumin is so much exhaustively studied. Anybody and everybody is studying about curcumin as anti-cancer drug. But if you remove curcumin from haldi and the remaining haldi extract was seen to show anti-cancer effect, means like Dr. Shirsagar rightly said, nature has given us in abundance. We just have to explore it. So this is a chance. And now some people have already also started this supercritical extraction. In supercritical extraction, what happens? We don't only isolate that one part of it. Curcumin is not isolated from haldi. We don't separate curcumin from haldi because isolation is not herbal approach. Isolation is chemical approach. In my opinion, people may differ with this opinion, but I believe that no Ayurveda is complete without that holistic approach. I mean, you cannot remove one part of the herb and say that this is that herb. That identity is lost completely. Also, there are opportunities of extraction, herbal extraction. You can do aqueous extraction, you can do hydroalcoholic extraction, supercritical extraction, that is the CO2 extraction, as I told you. After single herbs, there is also polyherbal formulation approach. Means, like two examples that we shared in our introduction, sugar regulating formula. Sugar regulating formula means there are at least 6 different herbs in this formulation, which act synergistically, and this synergisty works just magically in Ayurveda. So one of the example was sugar regulating. Then another example was turmeric related. Turmeric till now is known on, the most popular uses of cancer, anti-cancer activity. But we have used turmeric for peaceful, mindful sleeping activity. It works beautifully. in combination with some other herbs. So, my dear friends, please try to understand the scope, the size of the industry. The limit is no limit. Another thing is standardization. Standardization becomes a little bit technical. Technical means people who really like analytical studies, they go for standardization. Uh, Standardization means there can be different types of standardization. For example, you can do standardization of herbs. You can put up a lab where you can have reference herbs, and then uh, sta- uh, get these herbs standardized and prove that yes, this is the right one, this is the wrong one. So that is also a part. Again, if you talk about formulations, these formulations can be standardized. The process, the Ayurvedic herbal processing units work with some typical processes. So these processes can also be standardized. So there is again huge scope in standardization. uh, then there are animal studies. What's happening? What is this? Okay. Extraction. I already told you about supercritical extraction, aqueous extraction, hydroalcoholic extraction. Like I mentioned in our story, our sharing about in-house extraction facility, we do this extraction for our own herbal starting materials. Then I was talking about clinical and animal studies, like ashwagandha people have done. We also can do this. Like I talked about sugar regulating formula. So you can have tie up with institutes, hospitals, and people who can provide you with patients to perform these studies and prove the effect of your product. So this is like a life cycle of a herbal drug, or I don't like to call it a drug, but this is like a life cycle of herbal ingredient. Again, I think there is a mixed crowd of students in this audience. Maybe some people must be first year, second year, third year, final year pharmacist, post graduation students must also be there. So they must be feeling, the earlier students, the younger class must be thinking, why, what is of my interest here? So I would like to tell you this thing from the subject approach. There are different types of subjects, different subjects in our syllabus. I have just taken 4 examples just to show you the other side of it. Some people like chemistry, some people like pharmacognosy, some people like ecology, some are interested in analysis. So for everybody of you who is interested in different subjects, there are different business opportunities for you. If you can see the screen clearly, chemistry people go for standardization, they go for validation. Pharmacognosy people go for different types of farming. If they're interested in plants, herbs, and their characteristics and those things, they can definitely go for farming. Pharmacognosy people also are interested in polyherbal formulations or single drug formulations like ashwagandha, curcumin. And pharmacognosy people also would be interested in standardization, that is the fingerprinting and all. People who like pharmacology, there also we have good scope. We can do animal studies, we can do clinical studies. I think there was some pharmacology session in July from MET, similar kind of conference or sessions. So there only ecology approach was shown. I would like to tell you all that you can use that ecology approach on our herbal drugs, on our herbal ingredients, and definitely prove to the world what we are having. Then there are some people who really like analysis. So people who like analysis, there is analytic method validation, because there is really limited knowledge and information about analysis of herbal ingredients, analysis of polyherbal formulations. So for that, we really need to develop analytical methods. And in order to develop these analytical methods, you need that analytical mind. So people who are interested in analysis, they definitely can go for this. Again, standardization, it is everywhere. Also, there are some people who are not who don't like any subject in particular. But let me tell you, friends, this education is definitely useful. I'm not saying it is not useful, don't get me wrong, teachers. But having that business attitude, having that business acumen, if you have students who really have that business acumen but they're not interested in any subject, don't let them down. You can encourage even those people to become great herbal entrepreneurs. Not everybody who is successful in business is educated, definitely. So as a competitive advantage, if you say, you definitely will have that competitive advantage of being educated in the same field. So I don't know, I diverted somewhere else, but I feel this subject approach will be useful for young class of people. And you can see what kind of scope is there now. Again, I would like to connect this Ayurveda with the current situation. In this pandemic, the world has really realized the importance of Ayurveda. Everyone was consuming kada. Of course, some people did it over, excess. So it had side effects also. But I'm not talking about that. Anything in excess is poison. So we will talk only about generalized things. We also made kada. I would like to proudly share that we prepared our in-house kada, our in-house formulation, and gave it 1 cup every day to all our 300 people working. The whole year, the whole 1.5 year, everybody was working, everybody came to the factory every day. And till date, I am happy to share that everybody of them is healthy. Only 6 of them were affected by COVID. And that was too very mildly. They got recovered very fast. Means what we did, we prevented COVID-19 attack or maybe infection by using our Indian ancient wisdom. So by now the world already knows how we can prevent. Also, I know some Ayurvedic doctors who have cured COVID-19 patients only with Ayurvedic or herbal medicines. They have not taken a single chemical drug. Again, there were so many COVID patients who were treated with heavy antibiotics, antiviral drugs, which was, there was no alternative, and it was all a emergency situation. But post COVID, so many people were known to have side effects from these drugs, these antiviral strong drugs, and Ayurveda or herbal medicine has helped them recover from these side effects via these herbal immunomodulators. So we can imagine that the growth in this business can be much, much more than the estimated figures they have shown of that 550 billion or something. So I am damn sure that this business, the whole herbal industry, is going to grow much more than the estimation. Again, at the end, I would like to make it short. At the end, I would just like to share some real numbers with you all. Why real numbers? Because I don't want to boast about myself or my business or our growth, but this is because I want to show you the huge scope, the scope that I showed you in 550 billion in that global market. We have really faced it. We have experienced it. In 2009, we were only 1500 square feet. Today, we are another 1500 square feet, 50,000 square feet, and another 55,000 square feet facility is under construction, which is going to be live by 2021 end. So, and estimation of only Rave Pharma, only this small uh, company in the whole global business, by 2025 we plan to go to 1,20,000 square feet and 100 crores. These all are unimaginary but magical but real figures. And that's why I want to show you that, friends, there is no end to it. Even sky is not the limit. So you just have to start. Young budding entrepreneurs, the herbal industry is really waiting for you all. So many, after giving so many examples of scope and sharing my own experience, my own journey, I don't know how much it has been useful to you all, but maybe I would like to take a few questions.
Madam, with respect to Ayurvedic product, how regulatory agencies are looking towards this concept?
It is not clear, sir. Just a minute. Okay. Can I get the question again?
Yes, ma'am. Yes, ma'am. How regulatory agencies are monitoring these Ayurvedic products? Because we have
Okay,
seen.
regulatory agencies.
Yes, yes, yes.
See, first of all, in India, we are governed by FDA, Schedule T. Schedule T is the schedule under which FDA gives us guidelines to make manufacture Ayurvedic medicines. So we come under Ayurveda. If you go global WHO guidelines, there are WHO guidelines for herbal medicine, for herbal medicine manufacture and testing and regulate. All that regulation goes under WHO. There are a few countries, however, which don't identify. They don't know. They don't want to identify herbal drugs or Ayurvedic drugs. They don't call it drugs. Like I told you in the earlier example of states, in United States of America, Ayurveda is governed by USDA, that is the United States Department of Agriculture. Means they go under dietary supplements there. It is just as food. They don't recognize this as medicine. I hope I answered your question, sir.
Yes, 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 ma'am.
Okay.
Any other question from the audience?
Mr. Amit Roy had asked that quality control of raw material. I hope
Yes,
it is
yes.
answered in my session.
Audience, please post your question. Okay, one more question is that if the product is approved by FDA, say for example in
Okay.
Maharashtra, Ayurvedic product, so can we make changes in the composition of that product, or there is no need to go for
Can
again to get
your voice is breaking? Can we make changes in the?
already approved formula?
Of course not, sir. No, you cannot change, because if you have already approved that in India, even if it is Ayurvedic, the license number, even you get a different license number, even if you have different guidelines, but it is treated as an individual product. Means if you add turmeric 500 milligram and ashwagandha 20 milligram, and then
Yes.
you want to change, you saw that Arey, nahi, this effect is less, so I want to increase ashwagandha. You cannot do it on your own. By your own mind, you cannot do it. You have to get pre-approval, and only then you can. It is treated as good as drug.
Okay. Next question is, what is the minimum starting investment to start manufacturing?
Okay. Actually, there is a huge range of investment. It depends on what area, uh, where you want to specialize. If you're talking only about manufacturing of one section of maybe of, ointment or cream, one section, it will be minimum 25 lakhs of rupees, minimum, to start with. Means सिर्फ उसका approval ले लिया to start with, but limited investment, limited returns. So there are costs involved in building equipment. This will be 25 lakh. You will need a building of at least 1500 square feet to start with, so that it is approved by the Indian FDA. But if you talk about equipment you can get, like we started with second hand equipment, which was, I think, not more than 10 lakhs. Yes, but this is an old story now, 20 years back.
Yes. Yes.
And in 25 lakhs you can cover one section, maybe tablet, not tablet, it is a complicated and expensive affair, but you can do syrup or oil or creams, ointments, external preparation.
Another question is,
I hope I
apart from,
answered your question.
yes, madam.
Okay.
Apart from ashwagandha, in which herbal drugs we can go for import and export?
Your voice is breaking. Apart from ashwagandha,
Uh, in which herbal drugs we can go for import and export?
import and export, as I told you, there are lot many herbs to discover or explore. If you ask me, the local herbs that we have in and around us, that has good scope in abroad. Why? Because they don't grow it locally. And if you I mean, like our grandparents used to say, they have just neem ka juice, neem ke patte ka juice. It is not there outside. So if you just explore neem, because neem as a whole plant is also rich in active ingredients. Even its bark is different active, its leaves are different active. Again, there is amla. Then there is punarnava. Punarnava, the name itself says that it regenerates. Punarnava, it gives its life to the cells again. Means that is also an interesting herb. Again, ginger is an interesting herb. Why I was talking about Punarnava, I'll go to Punarnava back, because if it is going to rejuvenate all the cells, means it is going to help all the logic says it will help all the diseases. Again, there is gulvel. Gulvel means guduchi. बहुत सारे herbs है sir, so many of them. Especially I would just say that it is जितना local available है उस पे study करो और उसको market करो. That's what I can tell.
One more question is there. As you are dealing with herbal cosmetics, so one question is asked by the audience: how will you determine the safety and efficacy of cosmetic products?
Safety and efficacy of cosmetic products is determined same. It is there are no different guidelines for testing safety and efficacy of herbal cosmetics. It is treated same as the other cosmetics. In fact, only what we have to look closely at the other ingredients that we are using. Means if we are using aloe vera, what other excipients using with aloe vera? Is that interacting with your active ingredient? Also, there is this SH Kelkar, if you have heard, there is this lab in Mumbai who do allergy testing on instruments. They have instruments for allergy testing, so you can do allergy testing. That is enough.
Okay. My personal question
And
is there.
also, and also for the ingredients that we are using in this product, each ingredient has its own material safety data sheet, MSDS as we call. So that is also enough. Yes, sir. You
If
are asking something.
if we are developing anti-aging formulation, is it necessary
Okay.
for me to go for the different enzyme assay like SOD, catalase, which can prove the efficacy in human beings?
Okay. Now, this is a very deep question actually, and in testing of any activity of herbal formulation, there are different levels that we can do. Like these ashwagandha people did, you can do clinical testing on control group and non-control group, and after that, for mechanism of action, if you want to do the studies, yes, you have to do the enzyme activities. That is because it depends on what kind of uh, herb you are using, and what is the need for method development. I mean, what is the need to develop that method of mechanism of action, basically.
Oh, how to fix the combination? Okay. One more question: how to fix the
Have
combination
I
of different?
answered that question? Or maybe we can discuss in detail
Yes,
one to one
yes, sure, madam.
later on.
Yes, yes, yes.
Okay.
One more question is there: how to fix the combination of different herbal drugs in a single formulation? Means number of components.
Okay.
How to select the quantity?
Okay. Okay. Actually, we have a lot of Ayurvedic old herbal grantha. There are different,
Yes,
like, and there are different interpretations of these grantha. Like there is this Bhav Prakash. There are so many books. They mention these shloka. In each shlok, they talk about those ingredients which synergistically act on one particular disease or uh, mm, ac- one particular disease. Yeah, I can say disease condition. So that combination is defined by Ayurveda. That combination is decided by those shloka. They are in Sanskrit. We just need to translate and interpret it correctly to have that effect. There are books which have translated in Hindi, in Marathi. So those books, yes, they are very, very helpful in deciding the combinations. Only the interpretation is critical.
okay. So, uh, moving towards the concluding remarks of today's session, I would like to thank Anjali Kataria madam. She has explained different concept like documentation, how you can do documentation, standardization, and how can you start organic farming, vertical farming, what are the opportunities available in the context of single herb, polyherbal formulation, standardization, and the person from different areas like pharmaceutical chemistry, pharmaceutical analysis, pharmacology. And it was a nice interaction with you, madam, actually. So, on the behalf of Internal Quality Assurance Cell and Department of Pharmacy, I would like to thank you very much for your session, and you have spare your valuable time with us. Thank you, madam.
Thank you so much, sir. It was a great interaction, and I enjoyed also as well. In case student have any other questions, they can really email me. I will be happy to help, because as I told you in the introduction, I am really happy to see the interest in herbal industry
Yes,
of the college
definitely
and the
we will be
institute.
in contact.
Also, uh, also, if anybody is interested in putting up a setup, so Milind
Yes.
will be really happy to help.
Okay, madam.
You
Okay,
can
madam.
email me. I
Oh,
will
yes.
get back to you all, definitely. Thank you. It was a great
Thank you
session.
very much, madam. Nice. Thank you. Now, I request Dr. Sandeep Sonone, sir, to proceed with the next session.
Thank you, Dr. Patil, sir. Moving to the next session, we have with us Dr. Shishir Purushottam Pandey. Welcome, sir. Talking about this, sir, sir is a director, Ayurved Sanshadhan Vibhag, Ayurved Seva Sangh Nashik. He is honored by Maharshi Annasaheb Patwardhan, Vaidya Dr. D.B. Shende, रस औषधी पुरस्कार भाय वैद्य खादीवाले वैदिक संशोधन संस्था पुणे. Sir holds the membership of Atreya Pratishthan Aurangabad. In a number of research projects, he acts as a principal investigator. Some of his recent projects are AYRL capsule, then evaluation of efficacy and safety of isotonic plus eye drops in patients of immature cataract, then clinical evaluation of Zendu Chyawanprash as an immunity booster for its preventive effects of infectious in pandemic of COVID-19. He has also been involved as a clinical and preclinical investigator in number of research projects. He also held a position as coordinator Ayush faculty in Maha Arogya Shibir in 2017. In number of workshops and conferences, he is being honored as Ayurveda expert and resource person. Sir, we are very much glad to have you on this platform for this today's event. Now, let us have insights into Ayurvedic drug development by Shishir Pandey, sir. Over to you, sir. Thank you.
Thank you, sir. Thank you. Am I audible, sir?
Yes, sir, clearly.
Okay. Good morning, friends. Today's my topic is Ayurvedic drug development. I am thankful to the organizer to invite me for the session. I'm also, it's my privilege to invite you on the celebration of Azadi Ka Amrut Mahotsav as decided by our Ministry of Ayush. Before starting our topic, I would like to focus on the Ayurvedic concepts, and I will be focusing on preparation of medicine, standardization of medicine, and safety of Ayurvedic medicines in this lecture. There are four components of human health as described in Ayurvedic literature. The first one is doctor, the second one is medicine, third one is nursing care, and the fourth is patient. And it is seen that the medicine is the second top priority as far as Ayurvedic concepts are concerned. Then what is the definition of medicine as per Ayurvedic literature? As per Ayurvedic literature, anything which gives therapeutic effect, that may be the solid, liquid, and gaseous substances, even internal, topical, spiritual procedures that may heal the patient, even the assurance of the health may be called as medicine. The medicine which we call as a medicine, it should be able to prepare multi-dosage forms. It should have a multiple therapeutic properties. It should be highly potent and therapeutic, target specific, and without untoward effects. This is very important. and should have highly efficacy using at least the least therapeutic dose. By using least therapeutic dose, we must have high therapeutic efficacy. All of us know the definition of Ayurvedic drug as per Drug and Cosmetic Act. As per the Drug and Cosmetic Act, Ayurvedic, Siddha, and Unani drugs includes all medicines intended for internal or external use for or in the diagnosis, treatment, mitigation, or prevention of the disease or disorder in human beings or animals, and manufactured exclusively in accordance with the formula described in the authoritative books of Ayurvedic, Siddha, Unani, Tip, systems of medicine specified in the first schedule. What is first schedule? This is the first schedule. In the first schedule, there are 54 books included, and including the Ayurvedic Pharmacopoeia of India. What is the need of drug development in Ayurveda? The pharmaceutical industry is one of the biggest industries for economic development worldwide. Life is not easy for humans without medicine, particularly in the developed countries. Drug development has been a lengthy process and low rate of success and huge capital investment in it. On an average, it takes about 10 to 15 years for a newly synthesized compound to become a marketable therapeutic agent. Half of the drug candidates failed in the late stage of clinical trials, and soon after their approval, some new drugs have to be withdrawn from the market due to their severe side effects and clinical risks that are not detected in phase 3 trials. Many pharmaceutical companies are now focusing on the development of plant-derived drugs. Since human beings have evolved from simple herbivorous animals to omnivorous animals, the plant-derived herbs interact favorably with the human body and hence produce beneficial effect in terms of health promotion. Ayurvedic medicines are having huge literary support for its safety and efficacy, and it exists in the practice today. And comparatively, it is economical to develop the existing drug than to search for the new molecule. And that's why drug development in Ayurveda is preferable. What is the drug development in general? In general, a typical drug development process from herbal medicine broadly includes isolation of bioactive ingredients and synthetic modifications on it, then evaluation of safety and efficacy, then regulatory approval of the therapeutic agent in case of new drug, and the clinical trials. Then what are the components of Ayurvedic drug development? There are identification and prioritization of R&D needs. What is the basic need of the issue? Then literature survey regarding the issue. Then formulation of hypothetical rationale. Then initiation of drug development, that means quality control, quality assurance. Then preclinical safety, biological activity, stability studies with standard protocols, and development of integrated protocols for clinical trials, including GCP and traditional methods guidelines. Then clinical trials with standard procedure and with due approval of regulatory authorities. These are the components for Ayurvedic drug development. Before actual drug development, there is a preparatory phase in which prevalence survey should be done for which kind of health related issue we have to search a new medicine. Then the formulation of the drug, that may be single or combination drug, and the drug should be specific targeted indication or the activity. and it must be on the appropriate literature survey. Authentic support should be there for the further process of discovery. Then classical evidences are needed for the development of Ayurvedic drug development, and data of folklore claims are useful in the drug development in Ayurveda. Then raw materials, as per Ayurveda, there are so many raw materials mentioned in the text of Ayurveda. They can be divided into three parts: the plant origin, then the animal origin, and the mineral origin. There are three types of raw materials used in Ayurveda. Raw materials of plant origin, for the confirmation of medicinal plants, synonyms are used in Ayurveda. These synonyms are helping to character the plant, specific plant. These are similar to the taxonomy study. Say in Guduchi, Chakrika is the synonym. Regarding the Gokshur, there are three Kantak is the synonym. That means three horns are there on the seed. This will useful for the confirmation of the specific plant material. Then various parts are used as per the Ayurveda: leaves, stem, root, flower, fruit, seed, and sometimes all whole plant is used for the preparation of the medicine. And there is the scope for our faculty. What is the scope? There is a scope for confirmation of phytochemicals in different parts of the plant. Which part should be used for the development of specific medicine? It may help after the study. Then there are various parts used on the botanical terms. As far as botanical terms are concerned, aerial roots, aerial, bulb, dry fruit, dry seed, endosperm, exudate, extract, flower bud, flower petal, flower, fruit exudate, fruit pulp, fruit rind, fruit, gland, and hairs on fruit, gall, heartwood extract, heartwood, inflorescence, then latex, leaf pulp, leaf, oleoresin, pericarp, plant whole, then root bark, root stalk, root, rhizome, siliceous concretion, then seed, stem bark, stem extract, style and sigma, stamen, then sublimated extract, tender leaf, and wood extract. These are the parts generally used in Ayurveda as a medicine. There are 7 phases for the development of drug, any drug. I will discuss the procedure for Ayurvedic drug specially. In the first phase, there is drug development phase. Drug development phase, there are collection of raw drugs. The raw drugs may be collected considering the good agricultural practices as published by WHO. Then site selection should be confirmed, because the site selection should be selected on the basis of the soil, then atmosphere, then land preparation should be prepared as per the good agricultural practices. Then sowing and planting should be done as per the GAP guidelines. Then irrigation, weeding, then insect pests and diseases should be considered for the cultivation of the raw drugs and collection of the raw drugs. and finally harvest. These points should be considered while the collection of raw drugs. Then, good collection practices. Once we have decided the soil or the place from where it is collected, the collection practices includes the planning, where and when should be the raw drugs should be collected. Then collection time, specific time should be there. We can see the various raw drugs in the market, but it is collected before it's ripened. That should not be there, and therefore the planning should be there. The collection time should be specified, and collection sites. Then collection methods should be proper. Collection methods should be adopted while collecting the raw drugs, and GCP documentation is very, very important for the tracking. Then post-harvest processing, the collected raw drugs should be processed before using it in the medicine. It should be transported to the processing unit. Then processing site should be identified before that. And at that site, primary sorting should be done. It should be washed, dried, sorting and gradation should be is needed at the processing site. Then it should be packed, stored, and documentation and traceability during the post-harvest harvest processing is very, very important as far as drug development is concerned. As far as Ayurvedic texts are concerned, there are methods of collection and collection time and parts of medicinal plants. It is indicated in Ayurvedic text. Leaves and stem barks should be collected in the monsoon as per Ayurveda. That means in the mid of July to the mid of September. Roots should be collected in the spring. That means from the mid March to mid May. Flowers should be collected in the summer. That means from the mid of May to the mid of July. And here is the scope for our faculty members. What is the scope? There is a scope to study the status of phytochemicals on the climatic change. This topic is very, very, very interesting as far as research is concerned, especially in the Ayurveda. Some collection sites are mentioned which are to be avoided as per Ayurvedic text. Herbs growing on the dirty places should be avoided. The herbs or medicinal plants which are grown in the marshy land, burial ground, salty soil, then roadside, then affected by the season, which have been infected with worms, fire, and snow should be avoided. It is recorded that the collected medicinal plants from these sites will not yield the desired effects, and that's why these soils should be avoided. Then what are the nature of raw drugs used in Ayurveda? Generally, dry drugs are used, but it is mentioned in Ayurvedic text that the group of medicines, see, Guduchi, Kutaj. Guduchi means Tinospora cardifolia, Kutaj, Halarina antidysenterica, Vasa, Vasaka, Adatada Vasika, then Kushmanda, Shatavari, Ashwagandha, Sahachar, Shatapushpa, and Prasarini. This group should be used in the wet form, in the fresh, juicy form. As far as formulation is concerned, Ayurvedic formulation is concerned, there are guidelines that when we are using green fresh medicine for the preparation of Ayurvedic medicine, then it should be used twice as that of the proportion mentioned in the formulation. That means if we use dried amla in one part in the formulation, if we have to use fresh amla in the formulation, then it should be used twice in that of the proportion mentioned in the formulation. But these are the groups where no proportion change in this group as far as Ayurvedic formulation is concerned, and this group should be always used in the green and fresh juicy form. Then the next phase is botanical identification. In the botanical identification, pharmacognostic studies should be carried out. Then chemical studies basis on that their ingredients should be carried out. It should be based on the available guidelines. Chemical studies should be carried out on the available guidelines and classical methodology too. Ayurvedic Pharmacopoeia of India have done much work in this phase. Part 1, API Part 1 is dedicated for single drug monographs, and there are 9 volumes published till date. In volume 1, single herb drug, it is dedicated for single herb drug, and in volume 1 there are 80 herbs. In volume 2 there are 78 herbs. Then in 3, 100 herbs. In volume 4 there are 68 herbs mentioned in API. In volume 5 there are 92 herbs, and in volume 6 there are 87 herbs and 14 other materials in the volume 6. Volume 7 is dedicated for the minerals. There are 21 minerals mentioned in this volume. Then in volume 8 there are 60 types of extracts with microscopy and TLC. It is mentioned in Ayurvedic Pharmacopoeia of India. In volume 9 there are extracts. There are 45 extracts with microscopy and TLC. It is mentioned in API. This will help in the development of Ayurvedic formulation. Then various tests are applied for the Ayurvedic drugs. As per Ayurvedic Pharmacopoeia of India, the raw drug should be authenticated. Then foreign matter should be checked. Then organoleptic characters. Then macroscopic and microscopic characters should be applied. Then loss on drying, total ash, acid insoluble ash, extractable value. That means alcohol soluble extractive and water soluble extractive should be clearly mentioned before the standardization of Ayurvedic medicine. There are limit tests as per Ayurvedic Pharmacopoeia of India which are applicable to Ayurvedic drugs. Heavy metals, lead, cadmium, mercury and arsenic, there are limits. Lead should be maximum up to 10 ppm, then cadmium up to 0.3 ppm, mercury up to 1 ppm, and arsenic should be up to 3 ppm. These are allowed in the raw materials or even in the Ayurvedic medicine also. Then microbial load, the limits are prescribed in Ayurvedic ASU drugs. That means Ayurveda, Siddha and Unani drugs. As per their pharmacopoeias, Staphylococcus should be absent, Salmonella should be absent, Pseudomonas should be absent, E. coli should be absent, and total microbial plate count should be up to 105 per gram. And for topical use, it should be up to 107 per gram. Total yeast and mold should be up to 103 per gram. Then aflatoxins should be in this limit. B1 should be less than 2 ppb. 2 ppb particles per billion, not million. Then B1, B2, G1, G2 aflatoxin should be less than 5 ppb. Then chromatographic profiles should be there uh, using TLC, HPTLC or GC. Then SA markers compounds should be identified, and pesticide residue should be checked before using it in the formulation. Then in phase 3, preparation of medicine should be considered. For the preparation of medicine, formulation SOP should be confirmed. SOP for the preparation of the medicine, it should be standardized. Then standardization of raw drugs and standardization of process should be carried out. Then stability study should be carried out before the clinical trials or before the complete process of development. Then quality assurance of the drug. Then considering these should be considered, classical methods should be considered while testing the quality assurance, and current available physical, chemical, biological parameters should be used for the standardization. We will see what type of standardization is needed. There are classical drugs used as a raw or crude drugs mentioned in Ayurvedic text. There are so many extracts which is to be used in Ayurvedic preparation of the medicine. Then the extracts are aqueous extracts, generally aqueous extracts, sometimes alcoholic extracts, and hydroalcoholic extracts also. For the preparation of extract, there should have a SOP regarding the proportion of solvent, temperature of the solvent and extraction process, then the duration of the extraction, method of the extraction, filtration techniques, and preservation techniques. This should be carried out before the development. What test parameters are applied to the Ayurvedic raw drugs or the extracts? Total 21 test parameters are applied to the Ayurvedic drug before drug development. These are description of the formulation, then color, odor, taste, moisture content, total ash, acid insoluble ash, water soluble extractive, alcohol soluble extractive, pH, then bulk density, tap density, volatile content if present in that raw drug or in the medicine, then the residual solvent, chemical test for secondary metabolites, then TLC, HPTLC, HPLC with markers, then test for heavy metal or toxic metals, including lead, cadmium, mercury, and arsenic, as previously described. Then pesticide residue, microbial contamination, then test for specific pathogen, E. coli, salmonella, staphylococci, and pseudomonas, and aflatoxin. These are the test parameters which are applied to Ayurvedic raw drugs, Ayurvedic extracts, Ayurvedic medicines, as far as drug development is concerned. For compound formulations, there are so many compound formulations mentioned in Ayurvedic text. We will see one by one. First, Asava and Arishta. Asava and Arishtas are the medicines in which self-generated alcohol is generated. At the most, 10% alcohol is allowed. Then the source of sugar used for the Asava Arishta is jaggery or honey, and no cane sugar is used. Then the solvent used for the preparation of Asava Arishta are water or decoction of medicinal plants, juice of fruits or the part of medicinal plant. Then sugar percent is the sugar percent allowed is 50%, and sometimes above 50% are used for the preparation of Asava Arishtas. The test parameters, along with the previously said parameters, some additional parameters are applied on Asava Arishtas. These are optical rotation, viscosity, total solids, alcohol content, reducing sugar, non-reducing sugar, test for methanol, and total acidity. These parametric tests are applied for Asava and Aristage. For Arka preparations, Arka is nothing but the distillate of medicinal plants. Uh, these are applied for active ingredients. Uh, these are applied to the medicines in which active ingredients are in volatile nature. Uh, for the preparation of Arka, 1/3 part of the distillate is used as Arka and used for the therapeutic purpose. Then Avleha, Leha, and Pak is the dosage form mentioned in Ayurveda, which is in the combination formulation. In the for the preparation of Avleha, Leha, and Pak, semi-solid, it is the semi-solid preparation as in malt, and liquid parts or dosage forms are heated to become semi-solid, and sugars are mandatory for this for the preparation of avleha, and various types of there are various types depending upon the consistency. That means avleha, leha, prash, chavan prash, like that, khanda, etc. These are the types depending upon the consistency of the avleha, and which parameters are applied to the avleha along with the previous said: total solid content, fat content, reducing sugar, non-reducing sugar, then total sugars, SA for major ingredients or major constituents of the main ingredient are applied for avleha. Then for Google, there are Trifla Google, and so many Googles are available in the market. These are the formulation. Basically, Google is the exudate of Comnifera mucul. The tablet or pills are prepared using this content as the main ingredient as well as binding agent. And Google is processed with specified decoction of medicinal plants. Before it is not used directly in the medicine. It should be processed in specified medicinal decoction. Then other contents are added in the specified proportion, and the tablets and or pills are prepared, which we call it as Google preparations. There are parametric tests: loss on drying at 105 degrees Celsius, that means moisture content; friability test, hardness test, uniformity of the weight, and disintegration time. These are the parameters applied to Google preparations. For churna, what? For churna means the powder dosage form. Foreign matters, then powder microscopy, particle size is applied for the churna preparations. There are two types of churnas. Fine churna, that means churna of that means the powder of 80 to 100 mesh is used generally in the powder dosage form, and for the preparation of the decoction, that we call it as a quasi churna. The desirable particle size is 40 to 60 mesh. Then loss on drying at 105 degrees Celsius. Then total ash, acid insoluble ash, pH of 5% aqueous extract is applicable to the churna. Then water soluble extractive and alcohol soluble extractive. These parameters are applied to the churna dosage form. In Ayurveda, oil preparations are widely used for the therapeutic use. These are in the form of ghruta and tel. Ghruta means ghee, and tel means oil preparations. Pulp of medicinal plants, oil or ghee, and liquid dosage form of specific medicines in the proportion as prescribed in the formulation are mixed together, and it is heated to become moisture free. This is the ghruta or tel preparation. It is then filtered and used for therapeutic purpose. This is the formulation used in Ayurveda. Which type of parameters for the standardization are used for this dosage form? Rancidity, then specific gravity, pH value, loss on drying at 105 degrees Celsius, then congealing point, refractive index, then viscosity, iodine value, sap value, then unsaponifical matter, then acid value, peroxide value, free fatty acids, total fatty matter, and mineral oil test, which should be negative as far as Ayurveda and grit and tail preparations. These parameters are applied to the oil dosage form. Then there are lavana. Lavana is the dosage form. It is the peculiarity of Ayurvedic science. Some medicinal plants are mixed with rock salt in the specified proportions, and this mixture is placed in an earthen pot and heated in the gajaput, that is the furnace mentioned in Arabic text, the crude furnace, what we call. The burnt material is received and is used as therapeutic purpose. There are lavanas, narikel lavan, and so on. There are so many lavanas are mentioned in the Arabic text. For lavan preparation, total ash, acid insoluble ash, sodium percent, and pH of 10% aqueous solution is applied. Likewise, kshar is the peculiarity of kshar dosage form is the peculiarity of Ayurvedic system of medicine. For the preparation of kshar, dried parts of medicinal plants are burnt in open air to become white ash. Then the ash is then mixed with 10 times of the water. It is kept for the soaking. A clear water is obtained by separating residue. This water is heated to obtain its soluble content, and this is nothing but the kshar. For kshar preparations, acid insoluble ash, sodium, potassium, iron percent is applied, and pH of 10% aqueous solution is used for the testing of pH. For lepas are generally powders of ingredients derived from the plants or minerals. Some are solids, semi-solids, and waxy in nature. Powders may be fine or coarse depending upon the type of ingredients and the method of their applications on the affected part. Lepas are always to be applied externally. See, the parameters applied to the lepas are same that of the parameters applied to the churna. That means powder dosage. And for vati preparations, that means tablet form, powder ingredients from the formulation is levigated with a liquid dosage from the medicinal plants, and the granules are prepared from this mixture and is then compressed to become a vati or a tablet form. For the vati dosage form, loss on drying at 105 degrees Celsius, friability, hardness, uniformity of the weight, and dt is these are the parameters, disintegration time. These are the parameters applied for the vati preparations. Along with that, there are mineral and herbomineral formulations mentioned in Arabic text. These are purpati. Purpati dosage form, it is prepared by using shodhita mercury. It is purified mercury. Purified mercury is different from the shodhita mercury. And the shodhita sulfur. That means the process. There is some specified process for the preparation of mercury to be used in the formulation. Likewise, all these preparations are mentioned. These processes are mentioned in the Arabic text for specific metals and minerals. Then shodhita mercury and shodhita sulfur is triturated together to prepare black sulfide of mercury. Other specified ingredients are added to this mixture, and this mixture is melted in an iron pot previously lubricated with the ghee, and this melted material is then compressed between two cool surfaces to obtain thin hard film, what we call it as purpati dosage form. For the pishti, precious stones are used for the preparation of pishti. Precious stones are processed with specified material as mentioned earlier. It is then crushed to the fine powder. This fine powder is levigated with rose water and triturated in pestle to reduce the extra to the extra fine particle size, and what we call it as pishti. Then rasa yogas. These are nothing but the herbo-mineral combinations. Bhasmas along with kajali, that means the black sulfide of mercury, and sometimes without kajali, that means without mercury. These bhasmas are mixed with the powder of other medicinal plants as specified in the formulation. It is then levigated with liquid dosage form of specific plant material. Then it is triturated to become dry, and from this dried material, pills, tablets, and sometimes powder is used for the therapeutic purpose. These are the rasa yogas. These are the herbo-mineral combinations. Bhasma is the peculiarity of Ayurvedic text. What is Bhasma? Bhasma is prepared from the metals and minerals. Before directly using metals and minerals in the medicine, it is processed with some specified medicinal decoction of for some specified time. What we call it as Shodhan process. It is used to reduce toxicity and to prepare suitable form for the further process. Shodhan is necessary, and after Shodhan process, there is Maran process. What we call it as Murchana. This Shodhit or the processed metal or mineral is subjected to specific heating process in the furnace used for specific medicinal plants. Then the process is repeated till it passes through quality test. What quality tests are applied? I will show you the parametric test. The particle size become nearer to the nanoparticles by using this process. And here, after the process of Marana only, it becomes safe to use in the therapeutic purpose. What are the quality parameters for bhasmas? Total ash, acid insoluble ash, sulfated ash, water soluble extractive, alcohol soluble extractive, assay for elements used in as a raw material for the preparation of bhasmas. Then IR, XRF, XPS, XRD, SAME, XDS, AFM. These, as per the requirement, these techniques are to be used for the preparation of, for the standardization of bhasmas. As far as Ayurveda is concerned, there are Ayurvedic parameters also mentioned in the Ayurvedic text for the quality testing of bhasmas. These are Nishchandratva, that means it should be lusterless. Then Rekha Purnatva, then the bhasma should be fine enough to enter within the lines of the fingers. Then Varitaratva is the test, that means it should float on the water. Then Nirdhumatva, it should be smokeless. Nishwadu, that means the bhasma should be tasteless. And Apunarbhava, that means it is irreversible. That means by some specific procedures, it should not come in the previous form. Along with these formulations, some patent and proprietary drugs are used in Ayurveda as similar to the modern pharmacy. That means syrup, ointments, capsules, granules, confectionaries, then dusting powders, tablets, and suppositories. These are also used in Ayurvedic text. Then the fourth stage is preclinical safety studies. What type of preclinical safety studies are applied to Ayurvedic formulations? For oral formulations, single dose toxicity study is applied in mice and rats. Then repeated dose systematic toxicity studies should be applied for long-term, that means long-term toxicity studies in rats. Then reproductive and development toxicity studies are applied to some specific medicines for specific purposes. Then genotoxicity study, then carcinogenicity study is applied to Ayurvedic preparations for the drug development. And along with that, metal associated toxicity in case of any metal mineral used in the formulation should be applied before standardization. For topical preparations, dermal toxicity studies applied to Ayurvedic topical medicines. Then phytoallergy or dermal phototoxicity studies applied. Allergenicity or hypersensitivity in guinea pigs are applied for the topical preparations used in Ayurvedic system medicine. We will see the single dose acute toxicity study. Acute toxicity studies aim to determine toxic manifestations of test substances that occur when animals are exposed to one or more doses within 24 hours of period. What type of animal species are required? It should be carried out in 2 rodent species, mice and rats. Then both males and females are should be used for single dose toxicity study. At least 6 animals per sex should be used for this study. Dose levels and route of administration, the limit of 2 grams per kg or at least 10 times that of the intended clinical therapeutic dose should be used for this type of study, and should be used, the route of administration should be used same as recommended for the human beings. Then frequency of administration, 1 or more doses using during 24 hours period should be used for this study. And what are the study observations? Toxic signs and severity, onset, progression, and reversibility of the signs, and mortality, if any, should be observed, and should be observed for 14 days after the administration of the test compound. The observation should include the changes in the skin and fur, eyes and mucous membranes, respiratory, circulatory, autonomic and central nervous system, somatomotor activity and behavior pattern should be observed. Then observations include tremors, convulsions, salivation, diarrhea, lethargy, sleep, and coma. This should include in the during the observations. In repeated dose oral toxicity study, 2 mammalian species, of which one should be a non-rodent. Then both males and females should be used for this study. In case of rodents, each group should consist of at least 10 males and 10 females. And in case of non-rodents, each group should consist of at least 3 males and 3 females for this study. Route of administration should be the same as recommended in human beings. Dose levels, there are the study required to be done with 3 dose levels. First one is high dose level. It should the dose level should dose level that should produce observable toxicity. It is this we call it as high dose level. Then low dose levels should not cause any observable toxicity. That is low dose level. And within these dose levels, there are 3 dose levels should be used. And in addition, there are vehicle control group should be used for repeated dose oral toxicity study. See, period of exposure, what type of period should be exposed as per the intended therapeutic duration, depending upon the duration of proposed human administration. Period of exposure in the toxicity study varies. For in case of single dose in human dose, if a drug which is to be used in single dose in human beings, the period of exposure in the toxicity study should be 2 weeks, at least 2 weeks. For more than 2 weeks and less than 4 weeks in human beings, the toxicity study should be at least for 4 weeks. For the therapeutic, for the medicine used in the therapeutic dose, uh, within 4 weeks to 12 weeks in human beings, 12 weeks should be observed. 12 weeks should be the period used for this study. Then for the medicines used in 4 weeks, 12 weeks to 24 weeks in human beings, it should be studied for a minimum 24 weeks in animals. And for more than 24 weeks in human beings, the same should be expected period for the use of trial in animals. Then recovery phase, in order to investigate the recovery phase from the toxic changes, 50% of animals in each group are allowed to live for at least for 15 days, or varying the length of the time after cessation of the period of administration of the test substance should be used. Then what should be the observations for repeated dose toxicity study? These are the general signs. General signs include the monitoring of uh, home cage activity, that means behavior, conversions, biting, then locomotor activity, and so on. Then body weight, at least once a week before the start of the administration, body weight should be recorded, and once the test material administration is started, weekly once for the first 3 months, and followed by every 4 weeks in case of experimental duration more than 3 months should be recorded. Food intake measure it should be measured at least once a week before the start of the drug administration is initiated. Then clinical chemistry should be observed during repeated dose toxicity study. These are urine qualitative, appearance, color, glucose, bilirubin, ketone, specific gravity, blood, pH, protein, urobilinogen, nitrites, and leukocytes are tested as far as urine is concerned. And in general parameters, plasma glucose, serum albumin, globulin, total proteins are to be tested. As far as liver function tests are concerned, HUT, HGPT, AST, bilirubin, alkaline phosphate should be tested. For renal function test, blood urea, serum creatinine should be tested. As far as lipid profile is concerned, serum cholesterol, HDL, triglyceride should be tested, and sodium, potassium, and calcium electrolytes are to be tested as far as clinical chemistry is concerned. For hematological examination, hemoglobin, total, that means RBCs, hematocrit, then reticulate count, total WBC count, total differential white blood cell count, platelet count, terminal bone marrow examination, ESR, in case of non-rodents only, and general blood picture should be tested as far as hematological examination is concerned. Coagulation parameters are used as, in case of non-rodents, time bleeding, coagulation time, bleeding time and coagulation time, prothrombin time, activated partial thromboplastin time. These are applied to the non-rodents only. Other function tests are applied, say ECG, visual and auditory tests should be performed. For rodents, ophthalmological examination should be performed on a fixed number of animals from each group and at least once during the administration of the period. And for non-rodents, examination should be performed on all animals before the start of the drug administration and at least once during the period of administration. Then necropsy and histopathological examination should be done during this test. For this purpose, brain, cerebellum, and midbrain should be used. Spinal cord, eye, middle ear, then thyroid, parathyroid, spleen, lung, heart, aorta, esophagus, stomach, duodenum, jejunum, terminal ileum, then colon, rectum, liver, kidney, urinary bladder, epididymis, testes, ovary, uterus, skin, mammary glands, mesenteric lymph node, liver, kidney, testes, and uterus. These should be weighed for the study, and for parathyroid and rectum, these organs should be examined if indicated by the nature of the drug, only if it is mentioned in the therapeutic effect mentioned in the study drug. Then estimation of metals regarding the Bhasma preparations or herbomineral preparations, arsenic estimation, trace metals like zinc and copper estimation should be applied. Gold and mercury estimation is mandatory for Bhasma preparations or the formulations which are herbomineral type of the formulation. Then lead estimation is mandatory as far as herbomineral preparations are concerned. Then reproduction and developmental toxicity studies are needed. This should be carried out for all ASU drugs. ASU drugs means Ayurveda, Siddha, and Unani drugs containing crude drugs or aqueous drugs or hydroalcoholic extracts without textual rationale. Remember well, these are applied only for the formulations without textual rationale. That means these are not as per the text. Or any other solvent based on the extracts other than hydroalcoholic extract or alcoholic extract or other than aqueous extract. These tests are applied for such type of medicines. This should be proposed. The proposed study should be studied or used in women or of childbearing age. Say if the medicine is used to be in the age of childbearing age, then it should be tested for reproduction and developmental toxicity studies for the standardization. These studies are studied in 3 stages: segment 1, segments 2, and segment 3. Then special toxicity studies tests should be intended to be administered. These are intended to be administered for chronic illnesses or otherwise over a long period of time, say 6 months to 1 year. These are applicable for those medicines which are used for days together. Genotoxicity and carcinogenicity. Genotoxicity studies are conducted in vitro and in vivo to detect the compounds which induce genetic damage directly or indirectly. And carcinogenicity studies should be done in rodent species, preferably rat. And local toxicity tests are applied for topical preparations only. After that, the phase of preclinical efficacy and biological activity is studied after confirmation of the safety. For efficacy studies, preclinical efficacy studies, there is we should develop a model for that specific issue or for specific disease condition, and the same treatment should be adopted as in the safety studies are applied for preclinical efficacy studies. Then design of the study and formulation of the protocol. In this phase, a study should be designed, maybe the control study or a comparative study or a randomized study or open label study, sometimes blind study. The blind study includes single blind, double blind, and triple blind studies. Then phase 1 study, phase 2 study, phase 3 study, and phase 4 studies are designed for the clinical studies. And at last, execution of clinical trial based on GCP guidelines, good clinical practices. For the clinical studies, approval of institutional ethics committee or independent ethics committee is mandatory. Then CTRI registration is mandatory. Then after approval, this regulatory approval, the clinical trial is to be conducted. Then trial monitoring should be done. Trial coordination should be adopted, and data analysis and publication. Lastly, the publication is done on the basis of analysis during the clinical studies. Thank you.
Thank you, sir. There are few of the questions from audience. First one, what is the difference between Ayurvedic formulation and nutrapharmaceuticals?
Just a minute. Ayurvedic formulations are the formulations mentioned in Ayurvedic text as mentioned in the schedule first, and nutraceuticals are the formulations other than medicinal values.
Okay, sir. Okay, yes. Second one is what evaluation parameters are considered under the approval of any polyherbal Ayurvedic formulation?
Pardon, sir? Please repeat the question.
Yes, sir. Yes. So, what evaluation parameters are considered under approval of polyherbal Ayurvedic formulation?
I have already mentioned in my presentation, sir.
Yes,
for
sir. Yes.
the combination or combined formulations, the various parameters are applied as per the API, Ayurvedic Pharmacopoeia of India, and these are mandatory for the standardization of medicine.
Okay, sir. Okay, yes. Thank you, sir. Few of the questions from chat box. Can in Ayurveda, and it is be asked whether same designations are applied to such modified products.
Can we modify manufacturing methods prescribed in Ayurveda? Yes, of course. We can modify the formula or the dosage form. There are rules for the marketing and production and sale of such medicine. If the ingredients are same as mentioned in the text, then there is no need of any safety toxicity studies or any preclinical efficacy studies. If the raw materials are used same as in the formulation, but it is used for new indication, then only safety studies are not required, but preclinical efficacy studies are required. And after the completion of efficacy studies, these formulations are permitted for manufacture or for the sale.
Okay, sir. Another thing is that How can we control heavy metal content in Ayurvedic formulation during manufacturing?
As far as process standardization is concerned, if we adopt Ayurvedic concepts of medicine, we will control automatically the content of heavy metals in the formulation.
Okay, sir. Okay. Thank you, dear sir, for the excellent.
If we use shortcuts during the preparation of the medicine, definitely it will harm the formulation.
Okay, sir. Okay. Okay. Thank you very much, sir, for such a wonderful and in detail discussion on that the Ayurvedic formulation development as well as the evaluation of the most of that the Ayurvedic formulations. Thank you very much, sir. Thank you. Now I request Dr. Santosh Hajir, sir, to proceed with that the further session. Thank you.
Thank you, sir. Thank you.
Good afternoon. I welcome you all for national level one-day seminar on Herbal Drug Industry Mind to Project. And I'm more than happy to introduce one of a dynamic personality in the field, respected Dr. Sanjay Tamboli sir. Dr. Sanjay Tamboli sir is postgraduate MD in Ayurveda from the renowned Podar Medical College, Ayurveda, Mumbai. With a very strong academic background, sir has been always in the list of toppers. He stood first in All India PhD entrance. His extensive work on diabetes complication, diabetic retinopathy, has achieved award for best paper both at national and international level. Sir has also worked various research projects for renowned companies like Himalaya, uh, Raptocos, Brett and Company, Charak Sandhu. Sir is currently heading Abhinav Healthcare as a CEO of the company. Dr. Sanjay sir has been associated with Ayurveda research and manufacturing sector for the last 20 years. Through Target Institute of Medical Education and Research, that is TIMER, Dr. Sanjay sir has escorted the clinical research field in the Ayurveda by conducting more than 110 clinical trials on various Ayurvedic and nutraceutical products for renowned companies like Dabur, Marico, Imami, Alkem, Wellex Labs. These are the few to name. All these studies have been registered with the top registering bodies in India, that is Indian Council of Medical Research, ICMR. Sir has also published more than 20 research papers in the peer-reviewed journal of repute, both national and international, and as well as medical conferences. Sir is also having 2 patents to his credit. One is US patent and one is Indian patent. With this brief introduction and without wasting much time, I request sir to deliver his talk and guide the participant with your words of wisdom. Over to you, sir.
First of all, a very big thank to MET for this wonderful opportunity to interact with the students of pharmacy. I would have definitely loved to come physically to the place and then talk to students and meet everybody. But we all know these are challenging times, and we hope that this soon gets over, and then we probably next next time whenever we conduct this session, we meet personally and then interact with everybody. Having said that, however, best possible way we are able to communicate with each other through these new platforms, we wish to do that, and we continue to strive to educate, to get knowledge, and to like say bring about new ideas and new avenues for the upcoming budding students. So with this intention and with a big thank to all of you, I would like to start this presentation. I'll just share my screen. Can I request the organizers to share from their side?
Hello, sir. I have added from my side on the stream.
Okay. Okay. So everybody is able to see the screen, right? Is the screen visible to all of you?
Sir, it is not there in the presentation mode.
Okay. It is there. Okay, 1 minute.
Sir, you have shared window, I guess, but share presentation also.
Yeah, 1 minute. Are you able to see now?
No, sir.
Just a minute.
There is a fourth option, share.
Yeah, so I go on the fifth option. I put share link.
Right.
I need to click on the window. Okay, and then share. Is it visible now?
Yes, we can see the your computer, but not the presentation.
Now
it is
okay.
okay. Yes,
Okay,
it is okay.
Fine. So sorry for this glitch. So thanks for this background that you described about me. First to start with this whole presentation, first of all, I would like to mention a very important thing that many of you are doing currently your graduation or doing your post graduation, and some of you may be also associated with some of the other industry. What comes to our mind first of all when we see any product is that okay, how did this particular product travel this whole journey from being a simple raw material to a phase it has come down to being a product. So we need to very clearly understand this whole phenomena, and that phenomena can be understood from a very simple example that I'll give you. Like say, just imagine a Triphala powder that you are seeing in the market from any manufacturer, or a Leaf 52 kind of a tablet that you see from Himalaya in the market. So you see that on the label there are many things mentioned, that the composition is mentioned, the dosage form is mentioned, the indications are mentioned, that the way it is to be taken, the name of the products is mentioned. It is in different dosage forms like tablet, capsule, syrup, etc. So it travels this whole journey from being in the farms basically, or in the forest, to coming into your home as a tablet. So this journey could be understood in this one slide that you see in front of you. So imagine something that is coming in your mind, that okay, I want to develop as a researcher or as a product development guy. I want to make a product out of an ingredient. How do I make it? So what you'll have to do is you'll have to first churn your mind and find out that is there any traditional reference being mentioned about this particular ingredient that you are thinking of. First thing that you'll have to do it. Nowadays, because we have got a lot of connectivity through the web, and we are able to connect across the world. It's very easy to find out that okay, is there a mention of this particular ingredient or herb that you are thinking of? That's the first job that you will do it. number one. Number two is you have in your mind that okay, this particular product has got efficacy on a particular indication, but then what kind of research has been done or what kind of modern literature is available in terms of its constituents, in terms of its composition, in terms of its efficacy, safety, and etc. So that will be available in the modern literature research that you will have to do it. So these are the first two things that you will be carrying out whenever a product comes in your mind. So this concept of mind to product is this. The starting point is this, that okay, you churn out the traditional textbooks and then find it out that okay, where is this product mentioned in Ayurveda, where it finds reference in the Ayurveda books. And then once you are done with that, you can align with somebody who is an Ayurvedic expert and then talk to him about the product and all those things. Because many a times we find that when it comes to an Ayurveda ingredient, it is not consumed directly as it is available in the farm or in the forest. It has to be going through various processes, and therefore that process of what kind of process it will require, that will be available from the Ayurvedic expertise that you will be aligning with. Then what you did was you did a modern research, modern survey of literature, whatever is available, and try to find out the constituents, etc. Now, third thing is very important and very, very critical in this whole process is the experience. Now, what I mean by experience here is when you are devising any formulation, it has to be effective ultimately to the end consumer. Like say, for example, if you are making a laxative formulation, it has to be effective to produce like say laxation effect in a patient suffering from constipation. If it does not have that laxative effect, nobody is going to consume it. Now, where this knowledge is going to come from, or where this confidence is going to come from, is from the experience. Now, experience, when I mentioned here, is from two perspectives. Number one perspective is getting information from the already existing Ayurvedic experts or practitioners or medical doctors who very well know that this product is this particular ingredient, is it effective or not effective, or it causes problems, or at what dose is effective, something of that kind. So if you don't have that, and merely on the basis of traditional textbook research and merely on the research on the internet, you will not be able to devise any formulation. So experience becomes a very, very critical part. Just because you think that there is a book mentioning that this product is good, that will not create any good product basically. So you'll have to churn about the experience that is available on the product. Once you have done these three activities, the experience, the traditional knowledge, and the modern literature, then you will have to think over that, okay, I have now this ingredient with me. I have kind of decided as to what ingredients I'm going to put in the formulation. This is the three top points that I discussed with you. Then you come to a point where you decide that, okay, is this a good product to get license? So we call it as license qualification. So if the product is not good to get licensed, like say for example, if you come across some Korean kind of a herb, or you are searching on the internet and it's very good for, like say for example COVID only, you come to know that okay, there is a very good herb that is good in COVID, and then you find out modern literature also, everything you will find out. But then once you apply for a license, the licensing authority, or the one who gives you license, the FDA basically here, will say that okay, this does not qualify as a product in India. Then you are the whole hard work that you have done till this stage is all wasted. So it's very important to see what kind of license you are trying to qualify. Now, when I mean to say license qualification, it means, are you going to take this license as a modern medicine, as a pharmaceutical product? Or number one, number two, are you qualifying this as an Ayurvedic product? Number three, are you qualifying this as a cosmetic product? Or number four, you are qualifying this as a food supplement. So amongst these licenses, you'll have to first finalize that, okay, I want this license as Ayurveda product, or maybe pharma, or maybe Nutra, or whatever it is. Now these 4 points they don't require these 4 points. Till this point, you have not done any homework or any work as far as, like say, product manufacturing per se is concerned or development per se is concerned, these are all limited to either talking to people through experience or through literatures. Either it may be traditional or modern literature. So till this point you are all doing maybe like a theoretical work, I would say, till this point. Now from here onwards, from this point onwards, formulation, development, it's all more practical work. Now we come to a point where we have done that. Okay, I qualify this product as an Ayurveda product. I know that okay, this license could be possible. I have talked to like say experts, and they are okay with this product being licensed as an Ayurvedic product and all. Now let me start developing the product, or let me start writing the formulation. So once you are writing the formulation, you will have to first be very clear in your mind that is this formulation that I am making, this is going to be in which dosage form? Is it going to be in a solid dosage form, or a liquid dosage form, or a semi-solid dosage form? What is the dosage form that you are looking at? Because dosage form will ultimately decide your composition of the formulation, your specifications of the formulations, and subsequently how it is going to get absorbed in the body, how it is going to get excreted from the body, how at what dose it is going to be effective, etc. So whenever you are deciding on making a formulation, first and foremost thing is that you should work out as to what in what form I'm going to deliver this particular product. If you are not able to decide on the dosage form only, and then after doing all your hard work and you come to a point then saying अरे यार अच्छा होता कि यार इसको liquid form में बना लेते तो अच्छा होता, that is going to be very bad, because you'll have to again start from the scratch. So don't do that kind of an activity. Once you have decided on one dosage form, it's very simple from converting one dosage form into another dosage form. So once you have decided that okay, I'm going to make a tablet, and tomorrow you think that no, tablet के साथ में I also have to make a syrup also, or also I have to make a paste also. So that is very easy to do it. But if your starting point is not clear, then you are into a real problem. So you'll have to finalize the dosage form, number one. Number two is you will have to write a range as to, okay, I have decided that my product contains maybe 2 or 3 or 4 ingredients. Number one, it is in tablet form. Number two. Number three is I am looking at a dosage from this range to this range. Meaning, like I'm going to put ingredient 1 in this concentration, ingredient 2 in this concentration, ingredient 3 in this concentration, and there is a range that you will have to finalize. You cannot absolutely pinpoint it that okay, I am going to put 100 milligram or 120 milligram or something of that kind, because you are in that developmental phase. So what you do is you put a minimum dose and you put a maximum dose. Okay, so this is going to be the minimum therapeutic dose and this is going to be the maximum therapeutic dose. So between this is what the formulation will be developed at the final stage, is what you are looking at. Therefore, what is most important is that we devised the dosage form first, and then we thought that okay, these are 4 ingredients and each ingredient is to be put in a particular concentration. Now that concentration that we are putting is basis these first 3 things that we discussed. Okay, the traditional textbooks, the modern literature, the experience, and etc. So basis this three, you are developing this formulation and writing the minimum and the maximum range in which you want to make use of those ingredients. Now you have done that also, but then the question comes that okay, if I want to use, say, like for example, amla in the product. Now amla is a fruit, everybody knows it, but at the same time amla has got roots also, amla has got leaves also, amla has got stem also. So what part of the plant you are going to use it, that's again very critical. Why it is critical is because the active compound of that particular plant may be more in one part and less in the other part, or it is absolutely absent in one part. So that critical part you'll have to figure it out, that okay, when I'm decided that okay, I'm going to put amla in my formulation, amla fruit in my formulation. Now till that point you have arrived. Now again there will be a question that okay, I am putting amla as a fruit, but amla as a fruit is like fresh also and dried also. What I am going to put in my formulation? Am I going to make a powder out of fresh fruit, or am I going to make a powder out of the dry fruit, or am I going to churn it and then make it a very super fine powder, something of that kind? So the ingredients that you have selected, post that and then the range that you have selected. Post that, you'll also have to select that in which part of that plant I'm going to use in the formulation, and secondary, once I've done that also, and that specification of that ingredient is going to be what. Now these specifications will be determining the ultimate standardization of your finished product. So your raw material that you are putting into the formulation has to be absolutely very clearly defined, is what I mean to say. Many a times we find that in an Ayurvedic formulation, on the label, you just find, okay, this product contains maybe like 3, 4, 5, 10, 15 ingredients, but nothing is mentioned on the label as to which part of the plant is used, how did you standardize that ingredient, etc. These are all back-end work that are done at research and development levels. A good company would always test each and every raw material against some specifications, and those specifications are which has to be looked at when you are making the finished product. So this is the third thing that you look into the product development part, that is the specification of individual ingredients basis the part of the plant that is used. That's the other part of it. Then you have decided, okay, there are 3 ingredients, these are the specifications, etc. How you are going to now make the product as in, like say for example, I'll give you one more example. You decided that you are going to put, say for example, pudina in your formulation. Now pudina is available in the form of leaves also and in the form of crystals also. So we know very clearly menthol. Menthol is being derived from pudina. Then you decided that, okay, because leaves are very unstable kind of ingredient, they may like say when they are dried, fresh they smell very good, but when they are dried they lose their smell, and therefore we decided to put menthol crystals which are derived from menthol leaves basically. So now you cannot put like say 1 gram into the formulation, you have to put very low concentration. But then you came to a point that you decided that, okay, I am now going to make a formulation containing menthol crystals in a tablet form, but that menthol crystals are so unstable to heat that my process of product development has to be made in such a way that my ingredients in the formulations are not lost in the process, and therefore you will have to devise the process also properly. So that process is what we call it as product development process. So that critical part you'll have to define that, okay, at what temperature you are going to make the formulation, how you are going to mix it, how you are going to compress it, things like that. So once you have this formulation development itself is a very big topic by itself. So I've given you a broad idea about this. Now packaging development. You are done with the formulation, you have made tablets and everything. Now you decide that, okay, I want to pack this into either bottle. That bottle can be a glass bottle, that bottle can be a plastic bottle, that can be a blister, it can be in the form of pouch or anything. So different packaging materials are to be there. And why packaging materials are important is because they determine the stability of the product. Many a times what we find is that that product is good at the manufacturing level, but on the shelf when it is there in the market, it becomes bad, or it damages. Maybe the tablets become like say color, there is a change in the color of the tablets, or the tablet starts breaking, etc. Now the packing plays a very critical role over here, is because if you pack in materials which the product is not suitable to be packed, then you are into a big problem in spite of making a good formulation. So therefore you find many a times that there are in especially in pharmaceutical, you find very different innovative kind of packaging. Same applies for the Ayurvedic industry also. So you should see and check that is your formulation good or stable in your designed or your designated packing that you have decided. Now that decision of my product being stable is being done through stability studies. I'll not go into the detail of this. Just to give you a brief idea, stability study means to basically see that the product is stable over a period of this expiry date that you have mentioned. Say for example, you have mentioned 2 months, maybe 2 years, 6 months, maybe 3 years. Over this period, the product should retain all the qualities and all the quantitative specifications that are there. That for example, if I'm saying that my Tripla at 1 month is giving me effect for cleaning the stomach, it should give the same effect till the product expires. that is determined by the stability of that particular product. And that stability can be two types. Very broadly I can tell you is accelerated stability study and real-time stability study. Accelerated, in short, is something which we create an artificial environment to like say for the product to behave at a very at a little high temperature and a high humidity for a limited period of time. It can be 3 months, it can be 6 months. And real-time stability studies are in real world situations, as in like say on the shelf or on places where you will actually keep the product for that actual number of days for which you have mentioned the expiry. So here it will be stability. Then once you have done that also, then you will be testing this for efficacy. Efficacy screening means is okay, I have made the formulation, it is packed also, it is stable also, I have done a lot of work to make the product and everything. But at the end of the day, it should be effective also, and that efficacy testing also should be done. Now, these efficacy testings are done by way of clinical studies, preclinical studies, which Dr. Shishir sir has already explained in detail, so I'll not go into the details. Safety studies, basically why? Because we need to understand if the product that I'm taking, is it safe or not for that desired number of days, etc. Now, this whole process up to clinical studies is absolutely important for any product to be launched in the market. Good companies will always launch their products. Pharmaceutical, it is compulsory to launch only after clinical studies. Till this everything is done, only then you can launch a product. In Ayurvedic medicine, because some traditional knowledge is pre-existing, and you can catch hold of those pre-existing knowledge and then make a formulation based on that, and therefore it may not be mandatory. But now stringent laws are becoming more and more stringent. Safety studies are being demanded, efficacy studies are being demanded, and therefore you will have to it's a good practice to carry out these safety and clinical studies. Now, once you have done that, just as in modern medicine, now sometimes the FDA may ask you to make the application. At times they may not ask you. You can make direct application also. You make a label design, make certain claims on the label. Like say, if you see on the label there are claims like say for example, useful in constipation or useful in acidity. Now, these are called as claims, label claims basically. So your label claim is the primary indication of your product. Okay, so why you made this product? So that's how it is all about. So I tried to make this one slide explaining you in details the whole journey from traditional knowledge to label claims. Okay, so till that journey you will have to travel this. It's not very easy to travel this journey if you want to make an authentic, good, effective, safe product. Else you just make a product, take 10 herbs, just powder it and then pack it into a bottle and sell it, or maybe make a tablet and compress it and sell it in the market. But then, as a responsible scientific person, I think it's our responsibility to make products which are good, which are effective, and which are safe. So for that, you'll have to travel this journey. So this is what I wanted to communicate through this slide. Now, over here, this particular slide, I would like to emphasize upon some very important and very critical things. As in critical, why? Because Ayurvedic products many a times are looked from different angle because of their challenges, I would say. So the challenge, I have tried to enlist 3 primary challenges here. The formulation as a challenge, because you are fighting with using of crude material or extract material. That's the first thing that you will be absolutely very clear in your mind as to, are you going to use crude material or are you going to use extracted material? What kind of excipients are you going to use in the formulation, as in like the preservatives, the colors, flavors, binders, etc. This is a very, very important challenge that we find in Ayurvedic products. The other challenge that we discussed is the stability of the product. So I already mentioned you the real-time stability and the accelerated stability. So a real-time is 45, that is temperature and humidity. This is for 6 weeks to 2 years kind of a thing. Real-time stability at normal temperature and a low humidity, which is like say for a period of the expiry date of the product. The other important challenge that we come across in the Ayurvedic industry or Ayurvedic formulation development is the challenge with the heavy metals, challenge with the microbial load, challenge with pesticides, aflatoxins, and making the product standardized. Okay, so that for that we do a lot of fingerprinting, we identify certain markers. So I have tried to make you understand these three important challenges when you are making any Ayurvedic formulation. Rather than going into detail of this, keep in your mind very, very clearly in terms of when you are designing a formulation, what kind of material you are using, what kind of excipients you are using. That's number one. The formulation challenge which we discussed. And is my product stable over the expiry period that I mentioned? Many times we find out that we made a very good product, we packed it properly also, but after some days it starts having microbial load in the formulation, and then the whole goodness that you have created is all lost. At the same time, there are other challenges which I mentioned in terms of heavy metals, etc. So take care of these three important things, and you are done with your formulation. Now, this is a very exhaustive kind of table, which you should know it for sure, is because this gives you an idea of what is the permissible or permitted shelf life period for Ayurvedic ASU kind of medicines. I'll not go into the details. It ranges from, like, say, some months to a couple of years. Okay, so as a tablet, coated tablet can last for 3 years, or a capsule can last for 2 years. Oils can last for 6 months to 1 year, something of that kind. So again, this is very, very important and very critical. Now, one, because you are coming from a background of pharma, and therefore I need to throw some light on some very important factors when you are devising an Ayurvedic formulation. First thing that you will have to keep in your mind is cultivation and collection. Just as we have GMP and GLP, we have GAP also, which is good agricultural practices. So where you should know it very, very clearly that whatever source of material you are getting, is it from a farm, as it like some farmers have grown it, or is it coming from some collection like a forest? That determine, and the area from where it has come. Because since we are talking about natural ingredients, it is very important that we know that this Google that we are using, is it coming from Rajasthan, or is it coming from Maharashtra, or is it coming from some cultivated source down south, or a Tamil Nadu kind of a source? Because the active constituents will vary, and once the active constituents are different, the efficacy will vary, the quality standards will vary, etc. That's the first part. Then we come to identification of that material. Once you are testing any material for that matter, you should be very, very clear in terms of the macro and the micro analysis. The chromatography also is very, very important. Now, when I say chromatography here, without saying it includes TLC to HPTLC and GC, everything included in this. So identification is very, very critical. Heavy metal analysis by very upgraded methods. ICP-MS is one of the best methods that is being employed nowadays. Then you have pesticides, you have HPLC for testing aflatoxins, microbial count with the normal microbial content, etc. So whenever you are talking about Ayurveda products, if you think that Ayurvedic products are merely mixing of certain Ayurvedic herbs, it is a gross mistake that you are making. It is to be standardized, quality parameters have to be very stringent. Else the acceptability of Ayurvedic medicines will remain, or like say it will get confined to a particular area only. You will never become a global player, and therefore for your product to qualify as a global product, you should be very, very clear on all these quality aspects that we have highlighted over here. As far as heavy metals are concerned, so WHO has very clearly defined. Why I specifically highlighted this slide over here is because WHO for herbal products have very clearly defined heavy metal content. I'm talking about herbal products only. Don't think that these are applicable for herbo mineral formulations. Herbo minerals are completely different type of formulations. Okay, so for suppose if I'm talking for Triphala as such, or maybe Amla powder or Amla tablet as such, it has to qualify this basic standard of heavy metals. Else it will be a problem for you to send this to any country for that matter. So a 10 ppm of lead, 3 ppm of arsenic, etc. are very important. Now, when you are making an Ayurvedic product or an Ayurvedic formulation, these are some Bibles that you should refer to. Okay, so there are different volumes of API, of AFI, etc. So you should be very, very clear as to, and then these books have in detail mentioned about the ingredients and their profile, etc. There are certain other books also available which refer to the, like, say, quality standards of ASU medicines, etc., which you can use it. To your, for your knowledge, there is an American herbal pharmacopy also. Just as we have API, there is an American herbal pharmacopy also. There is a United States pharmacopy also, which mentions many of the herbs which are being used across the globe. Okay, so when it comes to safety, we have already stressed upon certain very important aspects, which I'm not going to again stress upon this, but these three are very important. The toxicity studies, Dr. Shishir sir already referred to the detailed on toxicity studies. It's very important that whenever we are talking about Ayurvedic formulations, we should do acute, subacute, and chronic toxicity studies and clinical studies to validate the efficacy and safety of our product. One very specific rule that you should keep into your mind is this particular rule, is the Schedule E1. Just as we have many herbs in the formulations, we also use certain herbs which constitute, like, say, in the group of poisonous drugs. Biba, Balatak, everybody knows this. If it is consumed internally in an unpurified form, unprocessed form, it can really create big problems. Okay. Similarly, there are ingredients like gunja, which is again a poison at a dose more than 5 grams at a time. So you should be very clear as to what ingredients you are using. Is it qualifying, or is it falling in that segment of Schedule E1 guidelines or not? If it is so, then you will have to be very, very clear. And important thing here I would like to state is that Schedule E1 herbs, if you are using into your formulations unknowingly, without mentioning it on the label, you are into a huge trouble actually. And you have to mention on the label to be taken under medical supervision only. Just as we have Schedule H for pharma, here we also have a Schedule E1 which is applicable for poisonous substances list. So that's very important. Efficacy, you can, as I mentioned earlier also, if the ingredients are mentioned in the Ayurvedic textbooks, AFI, API, etc., there are 54 classical Ayurvedic books apart from API and AFI, then you don't require them to be tested for their efficacy if they are used in the way it is mentioned in the books. But if you change the format, then you will have to carry out the preclinical and clinical studies. Preclinical can be in vitro, in vivo, and clinical studies should be done as per the ASU GCP guidelines, as per the ethical guidelines, etc., which are already laid down very clearly. Just to sum up this whole discussion today, what I've tried to again emphasize is on certain important challenges. Selection of proper raw materials, it's very, very critical, as we mentioned, because we are absolutely dependent on bioresources. The natural resources we are depending on, and therefore it's very important that we select the proper raw material. When I say selection, I also mean sustainability over here. Many a times what happens is we selected a material which is only available 100 kg in the world at a time, and then tomorrow it is going to get extinct, and then after 1 year, where you will get that material from? So sustainability and selection is very important. So you'll have to do that way. Proper manufacturing techniques in terms of temperature control, in terms of exposing them to certain processes. As I gave you an example of menthol, please remember this. Whenever you are handling especially volatile compounds, whenever you are handling oils and things like that, so that is very important. Meeting the quality standards, we discussed a lot on heavy metals, micro, etc. So that is again important. Sanitization and consistency of raw materials, very, very critical. Sanitization through marker identification, through consistency in those markers. And lastly, the establishment of safety and efficacy of the product. So this is all about this herbal formulation development from my side. I once again thank the organizers for giving this opportunity to discuss with you. I tried to complete it in the slot that was given to me, so that we can have some questions also. Thank you very much.
Uh, Thank you, sir, for a wonderful session. There is one question from the participant. You talked about artificial environment for stability testing. So, will you please elaborate it for some of the participants?
Yeah, so when we mean to say artificial environment, as in like you will have certain specific stability chambers. Now these stability chambers are designed in such a way that they provide high humidity, higher temperature, lower temperature. We call them as zones, because if you see globe as such, if you see the whole globe as such, you will find that there are countries where the temperatures are sub-zero kind of temperatures. You have -20, -50 degrees temperature also, and then you find deserts in other places where the temperatures are in the range of +50. At the same time, you have temperature humidities which range from 100% to maybe 10-20%. So what we do is basically in this, there are these specially constructed stability chambers. That is what we call it as artificial environment. And you have to keep them into those stability chambers and see periodically what is the change happening in them when you expose them to this higher or lower temperature or higher humidity, etc. So that's the whole thing.
Question from Ms. Himani Dave. How will you go for the standardization of a single chemical constituent present in the formulation despite it contains many primary and secondary metabolites?
Yeah, so it's a very, very good question, in fact. So when we say that when we are standardizing a product, what we are trying to do is we are creating a method where we are locating or we are identifying a marker which is not essentially the active marker. It can be just a biomarker. So I'll give you an example. For example, you take ashwagandha, and you know that ashwagandha may contain maybe 500 different kinds of constituents, and we then kind of we try to qualify them into different categories. We call alkaloids, and we call phenols, and then we call saponins, etc. But then this is a broad classification of that. But then if you identify that, okay, in ashwagandha there is an alkaloid called as withanol, then you have now identified ashwagandha per se, and then you quantify that also, that to the extent of, like, say 1% or 1.5% or something of that kind. So here you have arrived to a point where you are with confidence, you are telling that my ashwagandha contains 1.5% of ethanol, but that does not mean that the other 98.5% is useless. The 98.5% is also equally important. It may be more important than what we have identified. But what we have tried to do is we just try to identify one marker against which we are going to standardize this in that particular formulation. Somebody may make a formulation with another alkaloid, maybe alkaloid withanolide A, withanolide B, something of that kind, or maybe a vidaferin also. So these are just marker compounds to identify and to qualify and quantify a certain ingredient to make it standardized.
Next question from Mahale Tanuja. Which type of preliminary requirement to start our herbal small scale plant?
The preliminary requirement is intent. So if you intend and if you dream, nothing is impossible. So you just dream and intend. So that's the first requirement. Thereafter, things will fall in place. People will join you. You alone cannot do it. People will join you. Number one. Number two, I am not going to tell you that, okay, you require this much space and you don't require space, I'm telling you, because you can align with or join anybody who has got already a space. You can talk to people who are experts, so you need not have all the knowledge also. Most important thing is your intent of giving good quality product is what I mean. And thereafter things will fall in place, so don't worry about it.
So last question from Mr. Pawan. On what basis we select the multiple herbal drugs or ingredients to prepare the prepare or formulate the new formulation, what things mandatory to fix their quantity?
Yes. So the way we select a group of herbs in a polyherbal formulation is the outcome that we are looking at. I gave you an example of constipation. So when I'm looking at constipation, I will have to be very clear in my mind that okay, how I'm going to treat this constipation. Am I going to treat it with laxative, purgative? When I say laxative and purgative, am I going to treat it with bulk laxative? So when I say bulk laxative, then isabgol can be an ingredient. If I want to say purgative, then maybe something like a croton containing compound like say Jamal Gutta, we all know. So that kind of ingredient we are using. So it all depends on the outcome that you are looking at. Then once you have decided that, okay, this is going to be my outcome, thereafter what you do is you list down the herbs which give you that outcome. Okay, and thereafter you list down those ingredients and select those ingredients and see if there is a synergy between these ingredients. They should not be antagonistic to each other. They should be in synergy. That means they should contribute to your outcome. At the same time, many a times what ingredients that you select are not acting in synergy, but suppose somebody has got a bad effect, the other ingredient will nullify that bad effect. I'll give you another example. So I selected, say for example, senna, which is like a very good laxative. But the problem with senna is that it causes gripping. I don't want that gripping to happen. So what I will do is I'll put in senna along with ajwain. Okay, so then the gripping will not be there. So synergy at times, or at times removing the bad effects of a product can be the basis of selection of multiple ingredients.
Thank you, sir. On behalf of Mates Institute of Pharmacy, organizing committee of this seminar and all the participants, I acknowledge you sincere thanks for time sparing and guiding all the participants with words of wisdom and knowledge and very politely, sir. Thank you very much, sir. Now I request Professor Pratap Pawar, sir, for proceeding with next session.
Thank you, sir. It's my honor to represent the vote of thanks on the occasion of a national level one-day seminar on herbal drug industry mind to product, organized by IQAC Cell and Department of Pharmacognosy, Mumbai Educational Trust, Institute of Pharmacy, Bhujbal Knowledge City, Adgaon, Nasik. On the behalf of Department of Pharmacognosy, IOP and MET, I offer high gratitude to all the eminent speakers for sharing their knowledge and expertise in today's session. I am very much thankful to Honorable Sri Chhagan Bhujbal Sir, Chairman, Mumbai Educational Trust, respected trustees, Honorable Samir Bhujbal Sir and Honorable Pankaj Bhujbal Sir, Dr. Shefali Bhujbal Madam, for being our motivational support to conduct such programs and event for the well-being of students as well faculty. I extend my sincere thanks to HR Department, EDB Department, Marcom Department, Canteen and Disha Services for every help they extended for the successful conduct of the event. Last but not least, I am very much thankful to our team members, Mates Institute of Pharmacy, and respected Principal Dr. Sanjay Shirsagar Sir, all teaching and non-teaching staff members, students for every support and help associated with the program. With this concluding word, I here declare that the session is over. Thank you all of you. All the participants request to fill the feedback form. The link of feedback form is available in the chat box, and every participant will receive the certificate within the 2 or 3 working days. Thank you very much.